Hello, my name is Francisca Rodríguez-Cabello, virtual student reader at the Center for Biotechnology and Bioengineering at the Universidad de Chile. Today I will present the project called Design of Peptide with Desirable Activity Assisted by Machine Learning and Data Mining Strategies. To start, peptides are protein structure composed mainly of amino acids linked together by a peptide bond. One of the main problems of working with this type of structure lies in the high economic costs involved in its synthesis. Therefore, it is necessary to design and implement computational methods that facilitate the design of peptides with desirable activity. Here is the source of information on sequence comparison strategies using thermodynamic properties. Some steps are characterize and obtain distributional properties and characteristic by activity, compare distribution at level 1 using a statistical test, all versus all strategy, compare and do correlation, analyze at level 1, all versus all, analyze the results and determine relationship comparison, generate comparison matrices. The sequence strategy consists of the comparison of sequence by means of alignments. The steps are summarized in a script to align two sequences in the same family, obtain adjacency matrix and conservation distribution by activity, and three different scripts to align, compare distribution between groups, and analyze quartiles and conservation membership between sequence of different activities. Here, the comparison of sequence using coded representation, scripts distance through sequence in an activity, distance script 1 versus the rest, obtaining distance distribution. Modify alignment script to identify distance relationship. There are relationships between different properties. For example, there is a clear correlation between the properties of drug delivery peptides and transit peptides. However, sensory peptides are the only one that present only one dependence relationship, which greatly favors the design of this type of peptides when comparing thermodynamic properties. Neurological peptides show significant similarities at the phylogenetic level with sensory peptides, followed by drug delivery and immunological peptides. Remarkable, this type of approach facilitates the design and implementation of binary classification model of activity. Considering this, the concept of kinship between the analyzed sequence. Notoriously, unlike phylogenetic analysis, the numerical comparison shows relationship between neurological peptides and propeptides, which was not observed in phylogenetic analysis, and agreement was maintained with drug delivery peptides. In this way, it showed that the application of different methods to study the same problem facilitates the identification of different patterns that help to generate these rules. Using the recognized statistical rules, the classification system based on phylogenetic approach and the numerical distance methods, we design a methodology to create sequence with neurological activities. First, we define the rules. Next, apply a deep generative model. We create sequence. Next, we evaluate the sequence using the defined rules and check if proposed sequence is neurological or not. The iteration continues until stop conditions are met. We design and implement different computational methods to define the rules that characteristic the activities of a peptide. These rules are based on statistical, thermodynamic, phylogenetic, and numerical points of view. Additionally, we propose a design pipeline of peptides with neurological activity, allowing us to obtain 34 new peptides with same activity. Despite the great advance, it is necessary to generalize test rules to the different activity and develop systems that evaluate peptides with mood line characteristics and how to detect them. Acknowledgement to David, Kadina, Bruce, Alvaro, Medina-Ortiz, and Frances Arnold. I remain attentive to your questions. Thank you so much.